Glycogenolysis, meaning glycogen degradation. So in this video, we'll see how glycogen is degraded and it liberates free glucose molecules. So first enzyme which helps in glycogen degradation is glycogen phosphorylase. So glycogen phosphorylase, it removes glucose as glucose 1-phosphate from the glycogen. Now this process of removal of glucose as glucose 1-phosphate is called as phosphorylasis. Now this enzyme contains pyridoxal phosphate as the prosthetic group, and the alpha 1,4 linkages in the glycogen are cleaved by this enzyme. So this glycogen contains alpha 1,4 linkages in its straight chain, whereas alpha 1,6 linkage in its branched point. So this enzyme can specifically cleave only the alpha 1,4 linkages which are present in the straight chain. And it removes glucose 1-phosphate unit one at a time. Each glucose molecule is being removed in the form of glucose 1-phosphate, and that too one at a time. This enzyme sequentially hydrolyzes alpha 1,4 glycosidic linkage till it reaches a glucose residue that is 3 to 4 units away from the branch point. This cleavage or the breaking, this continues till it reaches a point where it is 3 units away from the branch point. The cleavage continuously occurs in the straight chain till this enzyme reaches a point where it is 3 units away from the branch point. Now this enzyme cannot attack the alpha 1,6 linkage at the branch point. This enzyme can only attack the alpha 1,4 linkage in the straight chain, and it cannot attack the alpha 1,6 linkage at the branch point. So because of the attacking of the alpha 1,4 linkage, the glycogen molecule, if it has n glucose residues, for each cleavage by the enzyme glycogen phosphorylase, there will be formation of glycogen with n-1 glucose residue, and that particular glucose is being liberated as glucose 1-phosphate. And if glycogen phosphorylase is only the enzyme that is acting on the glycogen molecule, the final product will be a highly branched molecule, and that is called as limit dextrin. Because this enzyme cannot act on the branch points, that is alpha 1,6 linkages, and if it is only acting on the glycogen, the result will be a highly branched molecule that is called as limit dextrin. Next, the enzyme which helps in glycogen degradation is, the process is called as debranching, and this debranching is done by 2 enzymes. Now we have seen that the glycogen phosphorylase cleaves that alpha 1,4 linkages in the straight chain, and this cleavage continues till it reaches a 3 unit or a trisaccharide unit away from the branch point. Now when it reaches this point, this particular 3 unit or the trisaccharide unit is transferred to the straight chain with the help of an enzyme called as alpha 1,4 to 1,6 glucon transferase. Now after this process of this transferring, once this trisaccharide unit is transferred to the straight chain, this particular branch point is free. Now this particular branch point is cleaved. This particular branch point is cleaved by the enzyme alpha 1,6 glucosidase. Alpha 1,6 glucosidase is the enzyme which cleaves the branch point, and this enzyme is otherwise called as debranching enzyme. One more name for the enzyme alpha 1,6 glucosidase is debranching enzyme, because it cleaves the alpha 1,6 glucose. Now this particular glucose, the glucose which is held to glycogen with the help of the alpha 1,6 linkage, is liberated as free glucose. So the point to be noted here is, when glycogen phosphorylase acts on the alpha 1,4 linkages, the glucose molecules are being liberated as glucose 1-phosphate. Whereas if the glucose molecule that is present at the branch point, when it is acted upon by the enzyme alpha 1,6 glucosidase, it is being liberated as free glucose. It is not being liberated as glucose 1-phosphate. These molecules are getting liberated as glucose 1-phosphate, whereas the branch point is being liberated as free glucose. So this is one point that needs to be noted in glycogen degradation. So the glucose 1-phosphate that is released by the action of the enzyme glycogen phosphorylase is further converted to glucose 6-phosphate by phosphoglucomutase. The glucose 1-phosphate that is released by the degradation of glycogen by the action of glycogen phosphorylase is further converted to glucose 6-phosphate by phosphoglucomutase. Now the fate of this glucose 6-phosphate in liver, what happens to this glucose 6-phosphate in liver is, in liver there is a special enzyme called as glucose 6-phosphatase. This is only present in liver and kidney. So this enzyme glucose 6-phosphatase hydrolyzes the glucose 6-phosphate to form free glucose. This free glucose is further released into the bloodstream. Next, the fate of this glucose 6-phosphate molecule in muscle. Now, when glycogen degradation occurs in muscle, the glucose 6-phosphate, what happens to this glucose 6-phosphate is, because the muscle does not contain this enzyme glucose 6-phosphatase. Glucose 6-phosphatase enzyme is absent in muscle. So, this glucose 6-phosphate undergoes glycolysis to produce ATP for muscle contraction. So, glucose 6-phosphate, this enters glycolytic pathway, further producing pyruvate. In addition, it also produces ATP, and this ATP is used for muscle contraction. So, that is the fate of glucose 6-phosphate in muscle, because this enzyme is absent in muscle. So to summarize, the key enzyme for glycogen degradation is glycogen phosphorylase. This glycogen phosphorylase only acts on the alpha 1,4 linkages, and this sequentially hydrolyzes till it reaches 3 units away from the branch point, and these glucose units are removed in the form of glucose 1-phosphate molecules. Now, once it reaches 3 units away from the branch point, this 3 unit, that is the trisaccharide unit, is transferred to the straight chain with the help of the enzyme alpha 1,4 to 1,6 glucon transferase. Next, because of the action of this transferring, the branch point is free. Now, this enzyme is this glucose unit, the branch point glucose, is acted upon by the enzyme, that is glycogen debranching enzyme, and this glycogen debranching enzyme removes the branch point glucose in the form of free glucose. So these glucose 1-phosphate molecules are further converted into glucose 6-phosphate by the enzyme phosphoglucomutase, and glucose 6-phosphate further it has 2 fates in liver and in muscle. In liver, because of the presence of the enzyme glucose 6-phosphatase, it is hydrolyzed to free glucose, which is further absorbed or released into the circulation. Whereas in muscle, because the enzyme glucose 6-phosphatase is absent, this glucose 6-phosphate enters glycolysis, through which it produces ATP that is used for muscle contraction. So this is briefly about glycogen degradation.